In this lesson, we'll be talking about endospore staining. So endospores are protective structures that bacteria will only produce when they're subjected to harsh environmental conditions. And by this, I mean nutrient depletion, lack of moisture, waste buildup, those types of things. And it's not something that all bacteria can do. Some bacteria, when you put them in a harsh environment, they will just die. But there are some, typically gram-positive, bacillus or rod-shaped bacteria that are able to produce endospores when they are subjected to harsh environments. And while with most plates that we work with in our course, or most cultures, while most of them tend to be fresh cultures, meaning we typically work with cultures that are about 24 hours old, when you want to do an endospore stain, you don't want a fresh culture. You want a culture that's been growing for typically 5+ days, because you want to deprive those bacteria of all the nutrients in the petri dish, and you want the toxic waste to build up. You want that environment to be harsh to force them into endospore production, because if they're in a good environment, they're going to remain in their active stage. So by subjecting them to a harsh environment, that forces them to undergo sporulation and then produce endospores. So typically for a successful endospore stain, you do have to have an older culture. When cells are in a sustainable environment, meaning they have nutrients and moisture, they are typically going to be in their active state, which we would refer to them as vegetative cells at that point. So here, these cells that are just all pink, these little pink rods, these are vegetative cells, meaning they're active, they're still in a good environment, have access to nutrients, so they're going to be actively growing and dividing. Now these little blue ovals here, those are free endospores that have been released from bacteria that were subjected to a harsh environment and now have produces endospore to remain in this dormant kind of protective state. So when they're in their endospore form, they are dormant. So they're not metabolically active, they're not growing and dividing. They are merely waiting it out, hoping that environment stabilizes again. When they're in a vegetative state, they are actively growing and dividing. So they are metabolically active, and they are able to continue to grow and divide as long as they have access to things like nutrients. So typically when a bacterium gets exposed to a harsh environment, if it's able to, it will induce a process in its cytoplasm called sporulation. So as long as it's in a good environment, it's going to be in its vegetative state and growing and dividing. But again, when it gets put into a harsh environment, it induces sporulation. Basically, it makes a copy of its genetic material, and then it encases that genetic material in a tough protein coat. This protein coat is actually made up of keratin, the same keratin that we have in our skin, but it forms this tough, durable layer around this genetic copy of the chromosome. So this protein coat that surrounds the genetic information forms this entire structure called the endospore. Initially, the endospore will start off inside of what we kind of call the mother cell, but then eventually, as the cell gets older, the cell will kind of burst and it will release just the free endospore. And this endospore, again, it's really just a protein coat that's surrounding the genetic material and protecting it from the harsh environment. And this endospore will just stay in this dormant form for days, weeks, potentially even months, and kind of wait for the environment to stabilize. At this point, it's dormant, it's not growing and dividing anymore, it's just waiting for the environment to kind of turn back around again. Should the environment become more sustainable again, meaning waste clears up, temperature stabilizes, moisture comes back, nutrients are available, should that happen, then the endospore will germinate, and the cell will go back into its vegetative form where it can continue to grow and divide. So with endospores, initially they do start off inside of the cell. Now eventually, once the culture has got significantly older, the endospores are released and they're just free endospores. But at the beginning, they do start off inside of the cell. So when they're inside of the cell, we can have three different positions. We can have central, which just means they're in the middle. We can have terminal, which means the endospores are all the way towards the end. And then we can also have subterminal, which means they're towards the middle, between the middle and the end. So this kind of right in between is central and terminal. We can also look at the shape of the endospore. So some are going to be perfectly circular, whereas some are going to be more oval in shape that we would call elliptical. And then sometimes the endospores fit perfectly into the bacterial cell and they do not cause any kind of deformities. Sometimes, because of the size of the endospore, they can cause the cell to kind of bulge out. But initially, these endospores do start off in the cell, but again, as the culture ages, those free endospores or free spores are released from the cells. And then just keep in mind, those are protective structures, those endospores. So they are just going to sit in that dormant period and honestly just wait for that environment to stabilize. And once it does, that's when germination kicks back in and the cells go back into the vegetative state. So they can start growing and dividing again. This is not something, again, that all bacteria are able to do. Again, it's typically gram-positive bacteria that do it, and it is typically the bacilli or the rod-shaped bacteria. These are just a few examples of bacteria that can do this. A lot of bacteria in the genus Bacillus and Clostridium are able to undergo endospore formation. So, for example, Bacillus anthracis, which causes anthrax, Clostridium tetani, which causes tetanus, and Clostridium botulinum, which causes botulism. So, for example, Clostridium tetani, its endospores are actually typically found in the soil. Clostridium tetani does not like oxygen, so when it's in the soil and exposed to oxygen, it's going to be in a harsh environment, so it's going to be in its endospore form. So what we find in the soil as far as tetanus is typically the Clostridium tetani endospores, which are dormant and can't really do anything to hurt you unless they get put into what they feel is a good environment where they could germinate and become active. And for them, a good environment is an environment with little to no oxygen. So typically what happens is someone gets a puncture wound. And that puncture wound, due to the damage to the tissue, has a low oxygen supply. So when Clostridium tetani endospores get into the puncture wound with limited oxygen, they're in a good environment, they germinate, they turn into vegetative cells and activate, and then they actually release toxins that cause violent muscle spasms. So not all bacteria can produce endospores, but definitely those that do can have some really dangerous features that we have to kind of look out for. So how we know if a bacteria can produce an endospore or not is we have to do an endospore stain. It is a differential staining technique, so it does require at least 2 dyes to stain different parts of the cell. So we can stain vegetative cells and then we can stain endospores. Again, we can even stain the endospores when they're still inside the cell. So typically we start off with malachite green as the primary stain. So you add malachite green, and this is typically done over steam. We like to do this over steam because the endospores are a tough, durable coat. So you do have to apply heat to kind of break it down just a little bit so that the endospores can absorb the stain. So typically you would have some boiling water, and then you would have kind of a mesh covering on top of the beaker so that you can set the slide that has already been smear prepped on top, and then you can add the malachite green typically for 5 minutes. And at that point, everything's going to be green, whether you're looking at endospores or vegetative cells, because that's the only stain we've used to that point. After that 5 minutes, you need to do the decolorization step. This one's really easy. All you have to do is rinse with water. So for the decolorization step with endospore staining, it is just a simple water rinse. What this is going to do is pull out all the green from anything that's not an endospore. So the endospores, whether they're in the cells or outside of the cells as free endospores, will all be green at this point, but any cells are going to be temporarily colorless. So then the last step is to counterstain with safranin. So safranin is typically applied to the slide, and it is going to stain those vegetative cells and the cells, the part of the cells that doesn't actually have the endospore. So typically you will see some cells, there's not pictured here, but typically you will see some cells that are just all pink, and those are just active cells. They're vegetative, they haven't produced endospores. In this image, we do see some cells that have produced endospores, so the endospores are still inside the cell, and the cell is then this pink part. And then we can see some that have subterminal, terminal arrangements, and we can even see kind of that elliptical or oval type appearance. And then we also see some free endospores here, so these ones have already been released from their mother cells, whereas in these the mother cell is still there in the pink, and then the endospores are in the center.